This PowerPoint covers the development of the cord in vertebra. But before we get started, I want to just review that at this stage, the development of somites have differentiated into sclerotome, myotome, and dermatome segments. During the 4th week of development, sclerotome cells on each side of the neurotube and notochord migrate around the structures to merge with each other. Sclerotome cells surrounding the notochord form the vertebral bodies, known as centrum in the embryo. Sclerotome cells surrounding the neurotube form the vertebral arches, called neural arch, in the embryo. Around the same time the sclerotome are forming primordial vertebra, spinal nerves are emerging from the neural tube, as you can see in blue here on the diagram. Development of matter. After the neural tube closes, neuroectoderm differentiates into a neuroepithelial tissue layer. Neuroepithelial cells or tissue give rise to neuroblast cells. Neuroblast cells become the cell bodies of neurons. Clusters of neuroblast cells form the primordial gray matter of the embryo. Nerve fibers, known as axons, emerging from the neuroblast cells are covered with myelin, and they form the white matter of the primordial embryo. Embryonic spinal cord. By the 6th week of development, the spinal cord has 2 clusters of gray matter formed by neuroblast cells. Neuroblast cells become the neurons within these clusters. The primordial gray matter alar plate has cell bodies that become interneurons, and the basal plate cell bodies become motor neurons. And as a review, neuroepithelial cells give rise to neuroblast cells, which give rise to the embryonic gray matter. Embryonic spinal cord. As development progresses, the basal plates expand, forming the H-shaped central gray matter of the embryonic spinal cord. At the same time, the sulcus limitans appears, and it actually defines the separation between the alar and basal plates. The neural tube eventually becomes the central canal of the mature spinal cord. Development of primordial spinal nerves. Bundles of motor neuron cell bodies in the basal plate give rise to axons. These motor axons exit the basal plate and spinal cord to form the ventral root of the spinal nerve. Bundles of sensory neuron cell bodies in the dorsal root ganglia give rise to axons as well. These sensory axons merge into the spinal cord to connect with the interneuron cell bodies in the alar plate. They form the dorsal root of the spinal nerve. And as a recap, primordial, motor, and sensory axons form primordial, ventral, and dorsal nerve roots, respectively, and they join together either proximal to the IVF or inside the IVS to form a primordial spinal nerve. Spinal nerve migration. Newly formed spinal nerves migrate out of the neural tube. Their goal is to connect with periaxial myotome and dermatomes. This migration cuts through the sclerotome, dividing them into a rostral portion and a caudal portion. And this initiates a process called resegmentation. Resegmentation occurs when the caudal half of a sclerotome fuses with the cephalic, aka rostral, half of an adjacent sclerotome. Once the 2 sclerotome halves fuse together, they form a precartilaginous vertebra. Formation of the intervertebral disc. The space created by the sclerotome separating during resegmentation fills with mesenchymal cells around the notochord. The mesenchymal cells are a primordial mix of fibroblast cells and extracellular matrix. Formation of the nucleus pulposa. The mesenchymal tissue that formed around the notochord forms the annulus fibrosus portion of the intervertebral disc, and eventually the notochord differentiates into the nucleus pulposus. Block vertebra is caused by improper segmentation of sclerotome. They didn't separate, and so that the two large sclerotome had fused together.